Thanks, Carrie, and thanks everyone for coming. I'm happy to be here. So today I'm going to talk about some of the work that I have been doing for really a long time. And so before I start, I really need to acknowledge that this is a huge collaborative effort that I'm going to be presenting. So work that I've been doing since 2007 with a variety of collaborators, several students, and those in bold are the main collaborators on this project. And so what we've been focused on is trying to understand how ecosystems have changed, how climate has changed in Eastern Africa in the early Miocene, and in particular, how that affects the evolution of apes. And so what I'm going to talk about today is a lot about vegetation, and so in particular talking about grasses. And so where we're going to start is way back in the Cretaceous. And so grasses first evolved on the Gondwanan continents in the latest Cretaceous. And so you can see here, these are some records of early grasses, in particular in India, are where some of the earliest grasses evolved. And so what we see are grasses evolve in the Cretaceous, and most of the records of early grasses actually come from things that are called phytoliths. And so these are little silica bodies that plants deposit within their cells when they're growing. And so we have this record of these early grasses. And then following the late Cretaceous, what we see is the beginning of the proliferation of grasses globally happens around, just after and just around the Cretaceous-Paleogene boundary in the early Paleogene. And so what we start to see is we begin to see grasses on the different continents. And so this is showing you different continents through time. And then beginning sort of in the late Paleogene, so in the Oligocene, maybe around between 30 and 40 million years, we start to see pretty broad expansion of C3 grasses, which suggests that this is really the onset of major open ecosystems that begin to happen. And then we see those ecosystems really dominating across all of the continents. And then finally, we see these things called C4 grasses occurring maybe as far back as about 30 million years, but really proliferating here in the late Neogene, maybe around 10 or so million years ago. And so I first want to kind of step back and give everyone a crash course in paleobotany and in botany and talk about, because I'm going to talk a lot about C3 and C4 plants. And so when we talk about C3 and C4 plants, what we're talking about is actually has to do with the photosynthetic pathway of these plants. And so you probably remember some of this from like high school biology or later on, but essentially the idea is that plants take in carbon dioxide and they take in oxygen through these pores called stomata. They then use an enzyme called rubisco to go through the Calvin-Benson cycle to make sugars. And so the thing about this is that photosynthesis evolved way back in the Precambrian, and it evolved back in a time when there was no oxygen in the atmosphere. And it turns out that rubisco has a really, really high affinity for oxygen as well as for CO2. And so one of the difficulties is if the plant takes in CO2 and it takes in oxygen, if it closes those pores, it closes those stomata, what begins to happen is the CO2 gets consumed, and then oxygen starts to build up, and then rubisco starts fixing with oxygen, and we get the photorespiration cycle. What the photorespiration cycle does is it consumes energy and doesn't produce sugars. It's a big negative for plants. And so now the issue, though, is that plants want to balance water loss, because these stomata, these are just big holes out into the atmosphere. And so if they keep their pores open all the time in times of water deficit, in dry environments when it's hot and dry, they're going to lose a lot of water. So they need to close their stomata. And so now the issue is that if you close your stomata and you do this typical photosynthesis, which is called C3 photosynthesis, you end up having photorespiration take place. So what plants have done is they've evolved a different way to do this, and they've evolved this more than 20 times in different groups. And essentially what plants do is they actually isolate rubisco and the Calvin-Benson cycle from the outside. And so what they do is they take in CO2 and they use an enzyme called Peppe, which makes a C4 acid, hence C4 photosynthesis, which passes across a membrane, which then splits and then fixes with rubisco to produce sugars. Now the disadvantage of C4 photosynthesis is it takes more energy. And so it's really only favored in a few different environments. And so the next step of this is then how this is manifest in the plants. And so I'm going to talk a lot about isotopes, and so just so everyone's on the same page, essentially what we can think about is in a C3 plant, plants can be more choosy as to the stable isotope they choose, carbon-12 versus carbon-13. They preferentially choose carbon-12, and then they have a isotopic ratio, which we then see in the organic matter of the plant. It's slightly different organic matter in the waxes, or or analcanes, the waxes on the leaves. When the plant dies, that organic matter and its isotopes get incorporated in the soil, and if they make pedogenic carbonates within the soil, that will reflect the isotopic distribution of those C3 plants. C4 plants, on the other hand, because they close their stomata, can't be as choosy. And so as a result, they end up taking up more of the carbon-13. They have a more positive or more enriched value, and there's about a 15 to 20 per mil offset between C3 and C4 plants. So more positive values are reflective of C4, more negative values of C3. Okay, so we've got our C3 and C4 plants, and now the next piece is when did C4 ecosystems evolve? And so there's been a lot of research that's been done that suggests, looking at a bunch of different isotope systems, that between about 5 and 10 million years, we see a major expansion of C4. So moving to the right means more C4. And so what we see is around this window of time globally, we see some expansion of C4 biomass. However, it's not really quite that simple. We actually see C4 evolving at different times. And so what this diagram here on the right is showing you is different records from different continents, from Europe and Asia, and from Africa and from South America. And so what we see is different timing of expansion, but we really truly see true C4 grasslands really show up at this window of time between about 5 and 10, but the first occurrence of them happens much earlier on the different continents. And so there's been a bunch of hypotheses for why C4 plants evolve. Things like maybe there's some CO2 threshold. So what this diagram is showing you is this is CO2 relative to daytime growing season temperature. So C4 plants are favored in warm environments, so they're often called warm season grasses, but they're also favored in times of low CO2. So it's been suggested maybe that there's a drop in CO2 that happens in the Neogene, and that corresponds to the evolution of C4. Maybe there's some interactions between plants and animals. C4 biomass tends to be favored in environments of high fire, and so maybe there's some fire threshold, or maybe there's some other climate or environmental threshold that happens in the Neogene, or just maybe it just happens for different reasons on different continents. And so my work has been focused in on Eastern Africa. And so Eastern Africa in the early Miocene, if we go from the Miocene around 23 million years to about 10 million years ago, there's a whole lot of stuff that happens. And so what happens in this time period is this is the transition from where we have the hothouse world of the Paleogene into eventually the icehouse world of the Neogene. We also see a bunch of things happening globally in plate tectonics. So we get the closure of the Peppe Sea. We have the acceleration, uplift of the Tibetan Plateau. At the same time in Eastern Africa, major rifting is happening, right? So we have rifting to create the eastern and western arms of the East African rift system. So this is creating topography in Eastern Africa. It's creating spatial heterogeneity on the landscapes. It's also a really interesting time in the evolution of early apes and primates. And so the earliest primates, we see them back here in the early Paleogene. We have New World monkeys and Old World monkeys evolving here in the late Eocene and Oligocene. And then in this time period, in the early to middle Miocene, this is where we see the evolution of our ancestors. The catarrhines, and then the hominoids, the early apes, the monkeys, and then eventually us way up here. So this time period where we go from the earliest C4 grasses to C4 grassland expansion, there's a lot of stuff that's happening. It's a really interesting period of time, both in terms of tectonics, regional and global climate, as well as evolution of early apes and other animals at the same time period. And so if we look more closely at Eastern Africa, what we see is a record that's a little bit confusing. And so one of the things that we see, so this record here that you're seeing, this is delta C13, and so this is leaf waxes from ocean cores, so off of the eastern coast of Africa. And what you see is very depleted values until around 10 million years, and then we get this expansion of C4 plants. So again, going back to this idea that maybe C4 ecosystems don't really show up until around 10 million years ago. But if we look at the terrestrial record, we see a little bit of a different story. And so what you can see here, again, remember more enriched, more C4, we have some evidence of C4 biomass as far back as about 16 million years ago. There's been a lot of argument about this, and so it's essentially kind of, people have suggested, well, maybe these are just a few sites, maybe they're not real, we don't know what's going on. And so in the absence of data, prior to around 10 million years ago, we essentially don't really know what ecosystems look like. And so this is a compilation of data. These are showing you different isotope systems. This is showing you phytolith record, and here this is the CO2 curve. And I just want to highlight the fact that there's essentially almost no data from around 10 million years to 25 million years ago. And so in the absence of this data, it's been suggested that maybe Africa was just a huge pan-equatorial rainforest, that the entire continent was covered by an equatorial rainforest, and these early apes were evolving in these forested habitats, and that's why they were successful. And then later on, there was some opening up of ecosystems in the late Miocene. And so this would be suggested here that maybe here around 10 to 12 million years ago is when we first see C4 biomass in Eastern Africa. And so this is kind of the motivation for our work, is trying to understand when did these C4 ecosystems evolve, or open ecosystems become important in Eastern Africa? And what were the regional patterns of C4 biomass in open ecosystems prior to the late Miocene? And when did C4 biomass first occur? And then finally, what are the implications for hominoid evolution? And so I'm going to show you some data that we've been working on over the last very long, almost 20 years, from a variety of early Miocene sites in Equatorial Eastern Africa, from Uganda and from Kenya, where we have a variety of data sets and also a really nice fossil record. And I want to start with by highlighting two of these sites. One of our oldest sites, this site called Moroto, and then a site that's sort of in the middle of the time period that has a long record on Rusinga Island. And so this site in Moroto is here in eastern Uganda. And it's a really interesting site because it has one of the earliest apes. It has this thing called Morodopithecus. And Morodopithecus is one of the oldest apes, and it has really interesting adaptations. And in particular, one of the things that it has is it has a femur and also vertebra that suggests that it had a somewhat upright posture. Not upright like us, but upright like a chimp, maybe, in the way that it moved. And so this is an adaptation that we don't see until much later. And so it exists in one of the earliest ape taxa. Some of the other things that it has are, it has the teeth that it has suggest that it was eating leaves rather than eating fruit. And the short limb anatomy suggests that this thing likely was a climber. It could probably move up and down through trees relatively easily. And so what we've done is we've looked at the site Moroto in a variety of different ways. And so one of the things that we did was look at the isotopes of the tooth enamel. And so here, this is the delta C13 of Morodopithecus. And then for comparison, there's a variety of ape and monkey taxa, as well as modern ecosystems and middle Miocene ecosystems. And the thing to take away from this is that the isotopes of Morodopithecus look more like baboons than they do like any modern apes, suggesting that this thing was perhaps eating things more like baboons, eating leaves rather than eating fruit. We then looked at the Moroto site in a variety of ways. We have a whole suite of data from the site where we collected data from paleosols. There's a whole thick fluvial succession at the Moroto site. And so what we see here, this is the stratigraphic column. And so what we see is when we look at the isotopes from soil organic matter, as well as N alkanes, as well as pedogenic carbonates, is we see really enriched values. suggesting the presence of C4 biomass at this 21-million-year-old site. Another thing that's really remarkable is this is the fossil layer in which we have Merodopithecus and we have phytoliths, those silica bodies that we see in grasses from that layer, and those silica bodies can be taxonomically informative, and in particular, we see silica bodies that are diagnostic of C4 grasses. So we have isotopic evidence for C4 plants, as well as fossil evidence for C4 plants, and then our climate reconstructions based on paleosols suggest relatively arid environments, precipitation values between about 500 and 1,000 parts per millimeters per year. And so if we put the entire ecosystem together of this site at Moroto, what we see is a very dynamic landscape in which we had ephemeral ponds and streams that was continuously changing and evolving, a very seasonal precipitation, a very highly seasonal environment with seasonal precipitation, and what we think is a woodland biome in which there was open parts of the landscape that were dominated by C4 grasses, other parts where maybe it was slightly more dense. And so what we suggest is that rather than this Merodopithecus evolving its traits for eating fruit in trees, instead what it's doing is it's using those adaptations to move up and down within trees and then across breaks in the canopy. And we would reconstruct the environment of Moroto at 21 million years ago to look something like this, perhaps somewhat analogous to the savanna chimps that we see in Western Africa, where they move across wide grasslands and in between patches of trees. So we have the site of Moroto that looks like a relatively open site with C4 biomass. And then I want to compare it to a site of Rasinga Island that's about 3 million years younger, in which we have a variety of different kinds of evidence. We have a very long record at this site. And so the site Rasinga Island is probably one of the best sites in Eastern Africa for understanding the evolution of early hominoids. It has an incredibly high diversity and abundance of primates, including like whole skeletons of many animals. And so we know a lot about the primates, and so what we've been doing over the last about 20 years is trying to reconstruct what the environments were at this site. And so I'm going to focus on a formation called the Hawegi Formation, and I'm going to focus on two different intervals within that. And so the first of these intervals is about 18.1 million years ago. And so at this interval of time, we have a variety of evidence from the site. So we have fossil leaves, we have sedimentology, we also have vertebrate fossils. And so what we see at this interval, 18.1 million years ago, is we have paleosols that are very poorly developed. And what that suggests is that there's not a lot of water moving through the system. We have a lot of evidence for times when we have high amounts of evaporation. So we have things like salt hoppers, we have mud cracks. I'm pointing to gypsum replacement. So these are like gypsum teepees that we'd see in an evaporated pond. So we have a pretty high evaporatic environment, suggesting times of the season with really high evaporation to precipitation ratios. Our fossil leaves suggest that our climate was really, really warm, but relatively dry. Also supporting those, that paleosol evidence, but also the plants, the morphology of the plants indicates that these were likely deciduous, meaning that they drop their leaves throughout the year. Again, something that we would expect in a seasonal environment. And so we would reconstruct this as this green square down here in sort of a woodland or perhaps at the very bounds of a tropical seasonal forest biome. And so this environment would have maybe looked something like this. Relatively dry environment throughout the year that had times of perhaps even bare landscape or that were highly evaporitic, and then maybe stands of trees around ephemeral ponds that existed for short amounts of time on the landscape. And then shifting about 300,000 years younger, we move to a time period at 17.8 million years, and we have a completely different ecosystem. This is a remarkable site in which we find fossil tree stump casts with roots pointing into it. We have fossil leaves sitting directly on top of it, and we also have the paleosol in which these trees were growing in. And I want to just stop by saying, I want to give a shout out to my PhD student, Lauren Michelle. And so Lauren went to the field in 2012, and she told me, I think I found these like tree stumps. And I told her like, I don't know, I think that's pretty unlikely. I don't think that should be a focus. We need to look more at the long-term record from this. And she's like, I really think this is key. And so we went back in 2013, and lo and behold, she found a fossil forest floor. So for the PhD students out there, don't always listen to your advisor. Sometimes they're morons. Because this is really a remarkable site where we found tree stumps, and not only did we find tree stumps, but we found them in the same stratigraphic layer over several hundred square meters. So we were actually able to map the distribution of trees on the landscape to reconstruct the forest structure. The fossil leaves indicate that it's a really warm and wet environment. The tree stump density indicated that we had a multi-storied closed canopy forest. The evidence from the paleosols indicated a whole lot of biological activity. We have here, these are like termite termitaria, we have orobatid mite fecal matter, all of this stuff indicating that this was a really biodiverse and really active environment, and one that was very wet and very warm. And so an environment that was fundamentally different from that environment 300,000 years earlier. Another thing that's really remarkable about this is despite the fact that it's in the same geographic location, we have fossil leaves from both of those sites, and they share one species in common. And so we see a fundamentally different environment, a fundamentally different ecosystem over this window of time. And another thing that's really cool about this site is that because we had so much of that layer preserved, we were able to look laterally across the landscape. And what we found was, in some places on the landscape, we found a high abundance of C4 grass phytoliths. And so what we would suggest is that this ecosystem looks something like this one here. This is Kakamega Forest in Kenya. And so this lighter green color, these are grassland breaks within a very, very dense closed canopy multi-storied forest. And so we would reconstruct this environment to look something like this, much more like something like this is Kabale National Park in Uganda. And so a fundamentally different environment. So if we think about this, the thing that's really interesting from the hominoid evolution piece is we have the same hominoid taxa in these different environments. So the same hominoid species were living in these very, very different environments over this window of time that we have preserved on Rasinga. And so if we go back to our original question, is that what were the regional patterns of vegetation in the early Miocene? There definitely is no evidence for a pan-equatorial forest. Instead, what we see is spatial and temporal heterogeneity in ecosystems. We have more open environments and more closed environments, and they're existing through time on the landscape. So then if we think about when did open habitats really become important, so again, we can go back to this record, and so we can look at all of these different sites here, and what we have is a variety of isotopic evidence from these different sites. And so what we see when we look at our isotopic evidence, so this is isotopic evidence from soil organic matter, this is from pedogenic carbonate. What we see is a fair amount of variability. So again, this would be C3 dominated, this would be pure C4 grasslands, and what you notice is a lot of these ecosystems, a lot of these locations have evidence for some proportion of C4 biomass. We see this especially strongly in the pedogenic carbonates. And so some of these pedogenic carbonates, we would reconstruct these as having maybe as much as like 80% C4 biomass on the landscape. And another thing that's kind of interesting is that we don't see a really clear trend through time. It's not like we start out closed and we move open. Some of the most open environments are the oldest sites in Moroto. And then we have very closed environments in some of the younger intervals of time. So there's no clear pattern through time. One of the things is, what are the pedogenic carbonate? Is this real data? And this is a question that's come up. And so we looked at the pedogenic carbonate in a variety of ways. And so I'm just showing you some of our data. And so there's a bunch of different types of carbonate that you can get. And so we have micrite microcrystalline, and we have sparry calcite. So it's often thought that sparry calcite is diagenetic. But we measured the isotopic signature of the micrite and sparrite and then mixed. And what we found is they had the exact same distributions. And this and a variety of other evidence gives us confidence that this data, this pedogenic carbonate data, is not the result of diagenesis. Instead, it's a real signal. and just that the pedogenic carbonate is telling us about a different part of the time in which the soil was forming, a more open interval of time compared to the soil organic matter. And then using a Monte Carlo model, so that my colleagues Bill Lukens and Dave Fox developed, which essentially is a way of assessing the distribution of the delta C13 values, and then trying to reconstruct what potential proportion of C4 biomass we had on the landscape. We did this for our different sites, and so essentially the way to interpret this diagram is 0, everything to the left of 0 indicates C3, everything to the right of 0 equals C4. The closer you are to the left, the closer you are to pure C3, the closer you are to the right, the closer you are to pure C4. And what you see is that there's a really significant evidence for C4 biomass at many of the sites that we have. We also have this evidence from plant waxes. So these are, this is N-alkane data. And so you can see here this N-alkane data. So here would be pure C3, over here would be pure C4. Anything in between would have some mixture of C3 and C4. I'm also showing you two different leaf wax systems. So two different chain lengths of leaf waxes, C31 and C35. And so the reason I'm showing you this is because C31 is produced by all higher plants, but C35 is preferentially produced by C4 plants. And so if the C31 and C35 isotopic signature are sitting on top of each other, that suggests that it's a pure C3 signature. If they're offset from each other, it suggests that there has to be some contribution of C4 biomass. And so here, as an example, this is the Moroto data, and what you'll notice is you've got the blue dots, which are the C31, and the orange dots, which are the C35, and the orange dots are all shifted to the right relative to their C31 counterpart, indicating C4 biomass contributing to this data. We see a similar pattern at Rasinga, where we also, I showed you some of that phytolith evidence. We also see a little bit of an offset at Buluk, and then similar small offset at Bukwa. So our N-alkane data is showing us the same pattern as the soil organic matter and the pedogenic carbonate, that we have a variety of habitats in which C4 biomass is contributing and is playing a significant and important role, and then also we have variability within the sites, that not every site looks exactly the same. And so if we put all of this evidence together, what this suggests is that we have a similar pattern in all of our isotopic systems. We have clear evidence for a non-zero fraction of C4 biomass at many of the sites. Essentially, C4 biomass is an important part of ecosystems in the early Miocene. And the other piece of this that's important is that open habitats were also very important parts of the early Miocene. And I also want to make the point again that we have early apes at all of these sites. So our ape taxa are living in all of these habitats, the very most open and the most closed environments. We have ape taxa in all of these environments. So if we then look at our fossil evidence from phytoliths, so these are actual pictures of some of the phytoliths that we see. So these, again, these are those little silica bodies that are deposited within plant cells when they're growing. And some of these are very diagnostic for C4 plants, such as these here. And so these are just different C4 phytoliths. And so what we see is a high abundance and diversity of C4 phytoliths through our record. And then what we were able to do is then reconstruct a maximum percentage C4. And so one of the things about phytoliths is phytoliths are produced more often by C4 plants than C3 plants. So our maximum percentage of C4 is probably an overestimate of C4 at our sites. But what we can see is at 3 of the sites, we see a maximum percentage of somewhere around the order of 20 to 40% C4 phytoliths, or C4 grasses. If we then look at an ordination, and so what this ordination is showing you is showing you different vegetation types, modern vegetation types. And so over here, these are evergreen forests here. In purple, these represent open forests. These are forests that have grasses within the forest system. And then over here on the right, these are grasslands and open habitats. And so the dark orange hull that we have here, this is our Miocene data. So our Miocene data overlaps with those open forests and then those really open environments, those wooded grasslands and brushlands and shrublands. And so just like our isotopic evidence, our phytolith evidence shows us that C4 environments, C4 grasses, and open environments were important parts of the landscape in the early Miocene. And so we would reconstruct our different environments of the early Miocene to look something like this. So all of these are the different sites that we have evidence for, and each of these little cartoons is a little diagram that I drew to suggest sort of what the environment would look like. And so what you can see is we have a whole host of environments. We have closed canopy forests, like what I showed you on Rasinga Island. We also have dry woodland to brushland environments with big breaks in the canopy. And in some of the most open environments, we actually maybe see wooded grasslands, like broad expanse of the landscape that are dominated by grasses with occasional trees on the in different parts of the landscape. And so another point is that we have this whole host of environments. We have environments that range from closed canopy forests to wooded grasslands, and we see them existing through the entire early Miocene. And I also want to point out that I've just arranged these randomly so that this figure looks nice. It's not based on the location at all, right? So it's not like an east to west, it's just look at it through time. But the point that I also want to highlight is that we've got like dry woodland and bushland environments around the same time as we have wooded grasslands, around the same time as we have closed canopy forests. Like there's no trend, there's no change that we see a trend through time. It's not like we're getting more open or more closed. There's just a lot of heterogeneity on the landscape. And so the answer to our second question would be, when did early habitats become important? And we'd argue that open habitats are common in the early Miocene. And so it's not a phenomenon of the late Miocene. It's something that occurs much earlier, 10 million years earlier than had been previously suggested. So what we can then do is we can take our data and we can then put it on that long-term curve of the record, the African record. And so here again, right, so we've got 25 million years ago to the present. And so, right, prior to our work, we had almost no data in here from about 20 to 25 million years ago. And so if we add our data to this record, we see we've filled in the record really considerably. But some of the points I want to make here, so this is soil organic matter, this is leaf waxes, this is pedogenic carbonate, and this is phytoliths, right? So what you see is this is a pure C3 signal, this is a pure C4 signal. Okay, so what our record shows is that in the early Miocene, in soil organic matter, in N-alkanes, and in pedogenic carbonate, we have a mixture of C3 and C4 on the landscape. Another thing that's really interesting is, aside from the pure C4 grasslands, which would be way over here, you can see it in pedogenic carbonate and in N-alkanes, and in the phytolith record, which we really don't see until maybe 3 or 4 million years ago, the distribution of isotopes that we see in the early Miocene is actually quite similar to what we see for the rest of the Neogene. And so what this is suggestive of is that C4 biomass became an important part of African environments as far back as the early Miocene, and it likely persisted through the Miocene and into the Pliocene all the way up to the present. And so another way of thinking about this is, what this is showing you is this is biomes. This is biomes in Africa today. And so they're color-coded. And so with the exception of these pure so the light gray here, with the exception of those pure C4 grasslands that we see today in Africa, all of the environments that exist in Africa today existed as far back as the early Miocene. Closed canopy forests, tropical seasonal forests, brushlands, shrublands, all those environments existed as far back as 20 million years ago, or 21 million years ago. And so really now the question is why? Why did this happen? And so if we go back to those hypotheses about why C4 grasses evolved, we can try to deconvolve this, at least for the African record. So the first of these is that maybe there's some CO2 threshold, that CO2 drops to a level in which C4 plants are favored. So what this diagram here, this is the recent record that was published, the Cenozoic CO2 record. Dan Brinker's part of that group, right? They published that long-term compilation. So if we look at CO2 in the early Miocene, it's somewhere between about 200 and 400, and so it's not really all that different than later on when we see C4 plants. So this being like some kind of drop or some threshold effect seems unlikely. If we go further back in time, the CO2 isn't significantly higher for a fair amount of time. So we don't think CO2 is the driver. In terms of this faunal vegetation interaction, maybe you get the evolution of animals that have a morphology that adapts them for eating grasses, and like it drives the evolution of C4. And we don't see that in the early Miocene. We don't really see any evidence for shifts in faunal vegetation interactions until much later in the Miocene, in the late Miocene. The same thing if we look at the fire record, although admittedly the fire record is not particularly good for Eastern Africa. We don't see any change in the fire record through really much of the Neogene, at least not until the Pliocene or the latest Miocene when we start seeing the C4 grasslands, so big expansion of fires. And so what we suggest instead is that perhaps the heterogeneity that was created on the landscape because of rifting, so this is the eastern arm of the East African rift system. This is the Nyanza rift, which was active in the early Miocene. The western arm of the rift is over here and it's active. That what this does is this creates spatial heterogeneity. It creates highlands and lowlands, and it creates microhabitats. And so these microhabitats and this ecological heterogeneity created the environments in which some environments favored C4 plants and others didn't. And so what we see is this rifting and tectonism maybe set the stage that allowed C4 plants to become locally abundant on the landscape. And so what we would argue then is that C4 biomass is an important part of African landscapes as far back as the early Miocene. And it was important locally, and it made an impact on local environments. So the final piece of this then is, what are the implications for hominoid evolution? I work with paleoanthropologists, and so we think about this, and they think about this, and so we talk about this. And so, so if we think about our early Miocene environments, we see this wide array of environments, ranging from these open landscapes with lots of grasses to ones in which we have high amounts of seasonality, so some trees in a somewhat open landscape, to closed canopy forests. And what's really interesting and remarkable is that our Miocene hominoids occur in all of these environments. And so all the evidence that I showed you is from Miocene hominoid fossil sites. And a great example of that is at that site over Singa Island. We have the same taxa, the same species existing in these very, very different environments. And so what this suggests is that early apes are biologically adaptable. Essentially, they can adapt to changing habitat and changing climate. And perhaps that's the reason that later hominoids become so successful. And the reason that this is sort of like a fundamental characteristic of all hominoids. Like, we live in all environments. Many apes can live in a variety of environments, and that may be why. This ability to be biologically adaptable to changing climate and changing environment. Another important thing is that it has been suggested, because of this concept of a Pan-African forest, that early apes were living in closed canopy forests, and they evolved their characteristics to eat fruit by climbing out to the very top edges of the canopy and grabbing fruit, and that they were evolved these morphologies for living in these multi-storied closed canopy environments. And so instead what we would suggest is, because of Moroto and some of the other sites, that rather than being a result of living in a closed canopy forest, the adaptations of early apes is the result of living in a heterogeneous landscape, in which these apes had to move up and down between trees and then reach out to the very edge of branches to eat leaves. And so these traits that allowed Morodopithecus to be successful in this environment are the same type of traits that allow apes to be successful in these types of environments. So these traits allow for that adaptability to multiple environments and are perhaps part of the reason why apes are successful. And it wasn't necessarily for fruit, it was this heterogeneity of environments that provided selection pressure to drive these adaptations. And so what we'd suggest then is the implications are that early apes evolved in multiple environments from very open to very closed, and that the ability to move up and down in trees and between trees and reach out to the very ends of the branches are what made them successful and able to be adaptable to a variety of different environments. And so if we put all of this together, if we put all this evidence together, what this suggests that in equatorial Africa, open habitats and seafloor biomass were really important parts of landscape in the early Miocene. So rather than this time period of the early Miocene being a closed Pan-African forest, instead it was a heterogeneous landscape in which open environments and closed environments coexisted, and that there was abundance of seafloor biomass at some of these sites. It also suggests that the African record of the earliest C4 is a similar age, or at least as old as the oldest records from North America. But again, still different from other places like in Southeast Asia and in South America. But these really pull back the evidence of C4 grasslands and C4 grasses being ecologically important. We see them on a lot of different places through time, suggesting that at least in Africa, that they're ecologically important for much of the Miocene. And then finally, if we think about the African record, early apes were impacted by this variety of environments, and that the mixture of closed and open environments was really key and important in driving the selection pressures necessary for the evolution of apes. And so what I just want to finish with is a little bit about where we go now. And so one of the things that we're doing now is we're thinking about trying to fill in that gap from 10 million years ago up here, where we've got good data, to our records that go from 17 to 21 million years ago. And so I have two projects that we're working on right here. These sites in Western Turkana that correspond, they're between about 14 and 13.8 million years ago, that correspond to this interval of time called the Miocene climate transition, when CO2 levels were dropping following the Miocene climatic optimum, and it's thought that this is where we had onset of northern hemisphere glaciation began perhaps at this time period. It definitely got cooler globally during this Miocene climate transition. And we also have a site from the Miocene climatic optimum, from the end of the Miocene climatic optimum, this site in the Koch. And so we have some data from these sites, not a lot of data, but a little bit of data. We also are continuing to work at some of these early Miocene sites to try to tease apart the record to better reconstruct landscapes. And so we have some new isotopic data. This data is like, the hot pink is like last week, hot off the press, hot off the mass spec. But what's really interesting is that when we look at this data from the middle Miocene, it looks a whole lot like the early Miocene. And so the isotopic variability in soil organic matter and in pedogenic carbonates is really similar to what we see in the early Miocene, suggesting that perhaps the environments weren't all that different in the middle Miocene than they were in the early Miocene. We also have phytolith data. So my PhD student, Benanzio Munyaka, is actually working on analyzing this phytolith data right now. And so this is an obvious glaring hole in our data set, and we hope to fill that in as we move through time. But another thing that's really interesting is if we look at climate reconstructions from the early Miocene, the Miocene climatic optimum, and that Miocene climatic transition. And so one of the things that we see is, so if we look at this diagram, so blue represents wetter than modern, yellow or sort of tan represents similar climatic conditions than modern, orange is drier, and white is we just don't know. And so if we look at the early Miocene, by and large, almost all of our data suggests that our sites were wetter than modern. If we look at the Miocene climatic optimum, so this is a time in which CO2 was perhaps similar to or even higher than today, what we see is that our precipitation estimates are either similar to or wetter than modern. And then in the late Miocene, we basically don't know. We have no idea. And so that's one of the things that we're trying to fill in. But this is a really interesting thing, right? Because remember I said that if we compare the environments of ecosystems that we see in the early Miocene, that they're very similar to what we see today, except for no full grasslands. And what we see in the Miocene, in the early and the middle Miocene, is the climate records suggest that it's wetter than modern everywhere. And so I don't have an explanation for this yet, but this is something that hopefully we can figure out. I'm hoping to maybe think about convincing Carrie and Ned to think way, way back in time, maybe. I don't know, there's something going on, right? There's something that's happening that's allowing these sites to be considerably wetter, and it probably has to do with global climate and where water is coming from and those kinds of things. And so with that, thank you very much. I really appreciate your attention, and I'd be happy to take any questions.
Students first. You asked me to do that. Go ahead, Dan.
You talk a lot about there being little grasslands in the forest, and pictures of modern forests and put it all together. You linked up with the presence of C4 grasses. Is the idea that the grasses pop up and then you're able to form the grasslands, or that something else has happened? You're there's holes in the forest and the grasses fill in, which goes first?
Yeah, so it's typically today in modern ecosystems, those are generally open parts of the landscape that are then filled in by grasses. A lot of in Eastern Africa, a lot of things that we see are the open parts of landscapes are actually controlled by forest elephants. And so the elephants are just they're big and they run through the environment and they knock down trees and they create an opening, and then because